In this third and final tutorial that will introduce us to the adaptive immune system, we're going to focus on the effector functions that lymphocytes develop that allow them to eradicate pathogens. So in the end of the last tutorial, we saw how T cells, when they become activated, can proliferate and form effector T cells that can migrate to inflamed tissues, or they can form other types of helper T cells that can migrate into the B cell zone and enhance B cell activation. So first I wanted to talk a little bit about these effector T cells and what are the types of functions that they develop. There are a number of helper T cell subsets that can develop after activation. And the type of subset that develops depends on the nature of the inflammatory signals that those T cells are exposed to when they're activated. And these T cell subsets develop very specialized effector functions that are finely tuned towards eradicating specific types of pathogens. So I have indicated here the three main helper T cell subsets: Th1 cells, Th2 cells, and Th17 cells, as well as the types of pathogens that they are good at eradicating. Now, at this juncture, I'm not particularly interested that you are able to recite or memorize these T cell subsets or the types of pathogens that they eradicate, but I wanted to put it up here as an illustration of the types of specialized functions that these helper T cells can develop. And these helper T cells will secrete cytokines that are really important for activating cells of the innate immune response and enhancing their ability to eradicate those pathogens at sites of infection. The other main subset of T cells are killer T cells, or CD8 positive T cells, and often these are referred to as CTLs, which stands for cytotoxic T lymphocytes. And as their name implies, these killer T cells are adept at finding and recognizing virally infected cells and killing them. And that means that CTLs, or killer T cells, are particularly important for eradicating viruses. So I wanted to give you one other example of how T helper cells enhance the activity of innate immune cells. And so here I'm going to focus on a T helper 1 cell. This T helper 1 cell produces a highly inflammatory cytokine called interferon gamma. And when interferon gamma activates or acts on macrophages, it enhances their activation. So here we have a macrophage in an inflamed tissue. It has been trying to kill pathogens that it has phagocytosed, but when it interacts with this Th1 cell and is exposed to interferon gamma being produced by that Th1 cell, this macrophage becomes highly activated. And when those inflammatory cytokines provided by the Th1 cell enhance macrophage activation, it will increase their antimicrobial function. So you can see this macrophage is now doing a much better job of destroying these pathogens within its phagolysosome, the pathogens that it is eaten up by phagocytosis. Cytotoxic T lymphocytes, on the other hand, are killers. And so this is just an example of a cytotoxic T lymphocyte that has found a virally infected epithelial cell, and it will secrete now cytotoxic granules that will perforate the membrane of this cytotoxic or this epithelial cell and lead to its cell death via apoptosis. This cytotoxic T lymphocyte, or killer T cell, will form what's called a cytoplasmic bridge with its target to be able to effectively secrete these granules that will then kill this target cell. Now, that's all I'm going to say about T cell activation and T cell effector functions. Again, T cells will develop specific effector functions that are targeted towards specific types of pathogens. B cells are activated in slightly different ways. They don't require dendritic cells to get activated, but rather B cells are activated by these free-floating antigens that are traveling down lymphatic vessels into the draining lymph node. What are the types of antigens that B cells that can activate B cells? Virtually anything. B cells can respond to lipids, sugars, nucleic acids, proteins, polysaccharides, all sorts of things. Protein antigens tend to induce the best antibody responses because proteins can also activate T helper cells, but really B cells can respond to most anything. Here's an example of a B cell that's been exposed to a virus, and this B cell is specific for a glycoprotein or a spike protein expressed on the surface of this virus. And when that happens, this B cell can undergo a round of limited clonal expansion, and some of these B cells will develop into a plasma cell. Plasma cells are antibody secreting factories. Often they're drawn with these stacks of Golgi because plasma cells really only do one thing, and that is to produce copious amounts of antibodies and secrete them constantly. When B cells get activated, they initially form these short-lived plasma cells that produce IgM antibodies primarily. However, B cells, when they recognize their antigens, will internalize that antigen and chop up that antigen, present peptides from that antigen on its MHC class II. And if you'll recall, I mentioned that some helper T cells migrate from T cell zones into the B cell zone, and this is where these T cells become important because they can interact with these B cells via their MHC class II, and they can secrete cytokines and provide other signals that enhance the activation of these B cells, leading to their clonal expansion and the formation of germinal centers. Within the germinal center, there's two really important things that happen. One is that many of these B cells will go on to form memory B cells. These memory B cells will stop secreting antibodies, and they'll just kind of sit around and they'll wait until they see the antigen again that activated them the first time. And when that happens, they undergo a rapid recall response that enhances the antibody response overall. And so the multiple exposures to antigens can lead to boosts in antibody titers, or the amount of antibody in circulation, because these memory B cells get reactivated, undergo another round of clonal expansion, and secrete more antibodies. The other thing that happens within germinal centers is the formation of long-lived plasma cells. These are plasma cells that can persist for years and continuously, over the course of years, pump out antibodies. These plasma cells are mostly IgG producers or IgA producers or IgE producers, and they will continuously secrete antibodies, and that's why you can have antibodies specific for a pathogen that you might have been exposed to several years ago. It's because these long-lived plasma cells are continuing to pump out antibodies specific for that pathogen. So what are the functions of these antibodies that are secreted? Well, importantly, antibodies mediate neutralization. And what that means is they can prevent infection or prevent colonization by binding directly to these pathogens. So here's a virus, for example, and these antibodies that are specific for the viral spike protein have blocked the ability of that viral spike protein to bind to its target cell and enter that target cell and infect it. And so this is called neutralization. A second way in which antibodies can lead to pathogen destruction is through a process called opsonization. Opsonization occurs when antibodies bind to the surface of a pathogen and then bind to a specific class of receptors called Fc receptors that are present on some phagocytes like macrophages. And this enhances the ability of macrophages to phagocytose whatever is in its environment. And so antibodies enhance phagocytosis through this process of opsonization. Think of it as like a salad dressing on a salad. It makes that bacteria much more palatable and appetizing to this macrophage. And lastly, antibodies can activate complement. We mentioned complement very briefly in the Intro to Immunology lecture. Complement proteins can mediate lysis of bacteria. Complement proteins can also be opsonins, just like antibodies can, and so they can enhance the ability of phagocytes to recognize and eat up bacteria. And lastly, we saw that complement fragments are really good at activating endothelial cells, inducing their vasodilation and enhancing inflammation. So antibodies participate in all three of these processes to really ramp up the antipathogen response. There are some other antibody effector functions listed here. Don't worry about them for now. If you've read about them or heard about them, we will address those later. Okay. Antibody responses, therefore, can be very long-lived. A single exposure to antigen with no T cell help can lead to the formation of short-lived plasma cells and a short-lived antibody response comprised mainly of IgM. But a single exposure to an antigen with T cell help can lead to the formation of long-lived plasma cells that can persist for quite a long time, in many cases for years, that can lead to long-lived IgG responses. And secondary exposures to antigens can reactivate memory B cells, leading to a second round of long-lived plasma cell formation. And this will boost antibody titers, it will boost the affinity of antibodies for their antigen, and it will again boost the number of memory B cells. The more times you are exposed to an antigen, the more opportunities your antibody response has to learn and to get better. And so the antibody response improves over time with exposures to antigen. Okay, that is all I've got for the adaptive immune response. Hopefully this will give you the foundation that you need to understand the impact of childhood vaccinations and how childhood vaccinations can provide protection from infectious diseases.